Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, this is another day for 12th class and what we are doing, we are studying about the molecular basis of inheritance. After really long, we are putting up this video, but I make sure that we will complete our syllabus by November anyhow. And after that, we will come up with something really cool for our channel. Meanwhile, if you are going through this chapter, we have already completed a bit of it. We reach to the part of the central dogma. Basically, we are seeing that how these molecules which we are studying, it's working in our body and how they are making the more things in our body to look like this what it is today. Starting with the basic things that the DNA contains all the information we know that, and every cell has DNA and how it is packed we have already learned it. In this video, we're going to be learning about that when the cell is dividing, means the bigger question to the Watson and Crick was that, okay, you tell about the structure of DNA. Now also tell that how every cell will have a DNA similar. Okay, so first of all, we till now know that the cell forms from pre-existing cells. Okay, in the 11th class, you might have studied this cell theory, which Rudolf Weischer gave. Okay, so it says that homogeneous cell e cellula means what? Cells are made from pre-existing cells. Now, from that concept only, if the cells are formed from pre-existing cells, means the first cell itself will gonna make the DNA. This understanding was there. But what was the problem? The problem was that if it has to make two different cells, means the DNA number should increase. Okay, it will increase. Okay, while increasing and while it is going to the next step, we need to know that how the DNA is increasing. That's the major question, that it increased also and it maintained its consistency. Means, for example, if I am here, you have to make one more copy of me so that you can put it in this room and in the next room. How you will do it? You will copy me to this. But how tough it is to copy me? Like, for example, if I'm standing here, can an artist make me? Can my sculpture be made like me? This is the question how they do it. So here what they do, here DNA, when this model was given by Watson and Crick, their model itself tells that they will be doing what? They will be doing the complementary base pairing. Means if A is there, it always be complemented by T. So if this condition was satisfied, means the concept of replication is also satisfied. So what it does, it says that the DNA will divide itself, giving you the easy peasy way to understand. So DNA will divide itself and its complementary pair will come up and make a new strand against the divided one. Means, for example, if I am the DNA and you have to make me, then what I will do, I will cut myself into half. And then with that half part, one half is here, one half is here, you are doing what? You are making the remaining part of it. So chances of becoming the same with respect to symmetrical alignments and everything would be higher, right? Same thing which happens with DNA. Means if this half part is there, its complementary will itself will come and join with the rest of the part of it. That's how the concept was given. Okay, Watson and Crick gave this concept. So we will say that the DNA replication is what? DNA replication is semi-conservatively. Semi-conservatively means that half of them in the next cell would be what? Preserve, conserve. Okay, so semi-conservative in nature, which I am explaining you over here, which means what? Means half of the DNA will be conserved in next cell. It won't matter that whether the cells are two different cells. One strand will go to the one, other will go to the other one, and its replicated form. But the question is where it is replicating. It's replicating within the parent cell only. That's why we used to say that the DNA content double in cell chapter. If you don't know, don't worry. When we'll be doing the cell division topic, these things will come back again. But if you have already done it, this thing would be much understandable to you. Okay, so if there's not, it's okay. During the revision time, go through with everything, go through with the 11th chapter from that when it's come up on this channel, most probably the next month. Okay, moving on means that DNA will be conserved in the next cell. So means half will go with the one, half will go with one. Along with that, a new one will be there, strand, which will be forming with it. Okay, now if this thing is there, okay, we know what, along with that, we also know that since base pairing was given in Watson and Crick model. So everything is understood over here. We have made it so that understanding is there. Then you will be able to do the statement based question in your board exams also, as well as in your NEET also, because assertion reason questions are coming up. Moving on, okay, this has been given, this has been told, but we need the experimental verification of the same. So who gave the experimental verification of them? These two cuties. Of course, not the cuties ones. These are the scientists or the researchers. And what are they? They are the Meselson and the Stahl. Okay, so we're going to be naming one as mentioned, one as the Stahl. So this is what? Meselson and Stahl. These two give what? These give the experimental verification for the semi-conservative. Can you see that they are using certain kind of test tubes over here? So this is only we're gonna be understanding. Okay, and this is what? This is the centrifuge. Okay, so machines, which is just making it separate out. Okay, what is happening? We will understand. So what they did? They gave what? Experimental proof for it. Experimental proof of semi-conservative nature of DNA. Nature of DNA. So what they did, thinking about them, their work. So when they were doing their work, they took what? They took E. coli. Now, you know from the part 1 of the chapter that DNA has what? DNA has the nitrogen present in it. So if I change the nitrogens in its atomic mass way, so means what we are doing? Instead of N14, I am taking N15 nitrogen. Means it's a little heavier in mass. Okay. But the charge is same. That means the neutrons will be increased on it. You might have studied this concept in the 9th class, right? So what they did, they grew E. coli. E. coli is what? It's a bacteria. It will have a DNA. Its DNA will also replicate when it has to form. So it's grew E. coli in N15 rich environment. In your NCRT, it's written something like N15HCl. Okay, so basically this is the compound where they have used. Now there was no other source of nitrogen except this N15. So what the bacteria do? They took the N15 ones and incorporated in its DNA and all the other nitrogen substances where it will be present, like its amino acids and all those things, right? So N15 is present in their body and they grew it for the several generations so that all of them has what? N15, or means the much probability is that only. Okay, you will understand this concept which I just said the line. Okay, for multiple generation, which incorporated the N15, that is what? Heavy isotope heavy isotope of nitrogen into their molecular of DNA, because DNA contains what? Nitrogen. Nitrogen is base, right? Okay, this is done. Now if the question would be that only the DNA will have N15 or the other, the answer is everything, because they have whatever the substance is abundant present around it, we're going to be using it for our daily life. This is the body adapt to the minimum requirements, or else the life will not sustain if it's very strict with it. Okay, so this happens. Now if you will put all of this bacteria into the density gradient, now it's a heavier one, so it will be what? It will, the DNA that will be present, that will comes what? At the very base of it. Means simple as what I did. For example, imagine if this is the test tube. I don't have a test tube right now, this is just a glycerine. Okay, so if this is the test tube, imagine, and I put this E. coli into it, I mixed it, okay, with what? With certain chemicals. Okay, it's not just the water, you can do it. Okay, in that because the density gradient can you only be fine. Density means what? How lighter or heavier with respect to certain substance something is, right? So I put it what? I put the cesium chloride into it, and I then put the DNA, and I then do this like this. I just mix it up with what? With a centrifugal machine. Okay, so what will happen? Everything which will be at certain density, they will mixed up. Now what will happen? The E. coli which containing N15, they comes at very base bottom. Okay, so for example, there is no pen. Okay, maybe there's a pen. Okay, I will give you examples. Also, this kit is of my brother, younger brother. He won't be able to say anything to me, but if the same thing would be done by my sibling, like the sister, he would have killed her. Yeah, that's how the relationship. Let's just see that. So what happened after this? If the weight is there, it will come up. So most of them get occupied at this level, so which is very closer to the base. Can you see the little bit of this is the marking? Okay, I don't have anything else to mark, so I mark it with the pencil. So here is what you will see, that lot of density is there, or lot of substance is there. Which substance would be here? There will be what? There will be a presence of the DNA over here. Okay, so we did that. We centrifuge it in the cesium chloride, then centrifuge it under cesium chloride solution, leads to, now someone will ask us why don't get to the bottom? Because of the density. Okay, because its density is that much only, it won't be completely submerged. So if you might have worked with the density of certain products, there is a difference between how they much will be denser compared to the other. The values are very different. Okay, so under cesium chlorides leads to assimilation. Leads to assimilation means it's getting accumulated. Okay, assimilation or accumulated is better word. Accumulated at certain level. Okay, so what it would be, like for example, if this is the one, this is your test tube, you have centrifuged it, everything has been done, this is the material present inside it. Till here it's completely filled. Okay, I'm gonna be making it filled. Okay, now in this, your DNA sample that you have given to me came somewhere over here. Okay, this is what? N14 DNA. Okay, now what they did, now E. coli takes 20 minutes to replicate. Okay, so they use this DNA, okay, and it lets to replicate. Sorry, not N14, N15 is there. Sorry. This is N15 heavy DNA. But now what they did, they put it for in a N14. Means N15 is removed and N14 solution has been given. Means 14 HCl is provided. Okay, so what's gonna happen now? Now they put it for 20 minutes for replication. Under N14HCl's solution. Means now we have changed it back to the lighter isotopes. Means changed environment to lighter isotope. So now what will happen? Now we will see that this DNA will replicate. And we found out what? We found out that there are two different gradients present now. So means what happened after this 20 minutes? After this 20 minutes, this whatever the amount of DNA would be present, means this N14. This N14 what is present over here, that has been there. This N15, sorry, there is what? Little upward. Like over here. What is happening? This is N14 and N15 DNA. Here it was what? N15-15. Both the strand of DNA was N15-15. Here it is what? N14 and 15. Means the layer at which it is coming up, it's little up. Because what happened basically, that now it's coming at this level. Earlier it was this level, now at this level. So basically what happened? Basically that if the things are lighter, they will be present little upper than the denser part. So we understood that what happened when the lighter isotope has been taken, then lighter isotope might have incorporated in it and it gives the light density compared to the heavier one. We understood this thing. Okay, let's do for 20 more minutes. Okay, means 40 minutes has been done till now. Okay, so now we are going what? We are going with 40 more minutes. So in NCRT what? Plus 20 from the last knot. It's giving that from the first 40 has been done. Okay, so we are taking plus 20 minutes. Total 40 minutes done. Now after this 40 minutes it's done, what they see that still they are putting it on N14 only. N14Cl solution only. Now what happened? Now there is what? There is a difference in the density again. And this time we see two different density gradients at two different levels. Nikki? तुम यहाँ पर हो? Okay. So one is at this level, means N14, N15. And along with that, there is a one more level, much lighter, N14, N14 DNA. Means after 40 minutes, we found this condition. Now, this is how it is going on. The major question is like how we evaluate this thing and understand in much different way. Okay, so means now before to go it, we gave certain name to these things. What are the names given to them? Now, number one is that over here, this is all what? Heavy DNA. Whereas this is what? Mixed DNA. So mixed thing we used to call it what? Hybrid. So this is hybrid DNA. But after 40 minutes, what we saw? We saw the hybrid DNA, means same bandwidth. The colors I have made it to show you it over here. Over there, it is what? Two levels. You can see that two different suspension in the solution would be there. Okay. And this is what? Light DNA. That has came into it. Let's try to correlate with respect to only one DNA. Okay, we will try to understand with the one DNA, one circle. So we building the concept over here. Okay, So initially what was there? There was heavy DNA only. Okay, so heavy DNA, we made it like that. So this is what? A DNA. Both I am seeing with the red means this is what? N15, N15. This is what? Heavy DNA. Now basically what is happening? You know that each strand will grow its own counterpart. That's how it's going to be making up to the next. So what happened? When you put it on the N15 solution, that 20 minutes, what happened? On the N14 solution, what will happen? This thing will change. Means what? There will be two DNA because only one DNA molecule is there. So it will be what? N15, N15. With this N15, what comes? This N14. N14. So one DNA will make two DNA. Okay, the increment is like that. Later on, what happened? We even continued this thing. Plus 40 minutes means plus, like now what happened? 20 minutes has been added. So means total 40 minutes is over. Keeping it N14 solution only. Now what's gonna happen? We have what? This is N15, N15. Okay, we have the strand of what? We have a strand of N14, N14. So this is what? N14, N14, N15 and N15. But on the top, what has been added up? There is what? N14 will be the counterpart of it. So this is N14 strand. So what happened? There is a strand of N14, N14. There is strand of hybrid also. So what happened? The amount doubles than the last. So means after each cycle, means what? After each replication cycle or the replication, the amount of DNA doubles, which used to say na, like in cell division chapter, amount of DNA doubles. So if someone will ask you like what is the amount of DNA that has been increased, that would be what? The 2 raised to power n. N is the number of cycles or times it has gone for the replication. Okay, so means that number of DNA is equals to 2 raised to power n after n number of cycle. Clear with this thing? We understood this. This will come up in the biotech also. Along with that, what else we understood? We understood that the hybrid DNA was there. Here, what is here? If you see over here, this is what? This is all in this phase over here. This is what? All of this is what? Hybrid DNA. So this is hybrid DNA. And one thing I have to write it. So I have to just change it a little. We will just write it over here. That is N15, N14. Okay. Now, if you see over here, this one and this one, this is what? This is similar like the last one. That is what? Hybrid DNA. Whereas this one and this one is what? They are the lighter DNA. So means after 40 minutes, there is what? 2 different types of DNA present and equal proportion at 40 minutes difference. These things should be in your markings. Okay. So it means what? Here we have what? Hybrid ratio light at 2:2. That will be what? 1:1. Equal proportion. But will this equal proportion will sustain in later on? Let's try it. So what? Extra 20 minutes we go on. Okay. So here what we're going to be doing? If this you understood now, you are able to do anything. Okay, so now what, 60 minutes are done, keeping it at N14 Cl solution. Now what will happen? N14 will incorporate in the new strands. So we already have what? We have already have total 4 DNA molecules, means total 8 strands. So now we will make individually 8 strands responding to this, what has been given to us. So what is given to us? We had been given with what? We had been given with that there is 2 N15 strand. So I have taken one strand from here, one from here. Okay, rest all the other like 1, 2, 3, 4, 5, 6. 6 strand is N14. Okay, so I'm going to be making the 6 strand with N14. Much easier for you. Just remember this trick. So 1, 2, 3, 4, 5, 6. So these are what? N14 strand. So I'm just keeping it with the colors. Now just remember the colors. Now since the molecule is what? Molecule is N14 only. N14 I'm showing with the red color only. So I will make the counter part of it. So this is what? The counter DNA part of it. Second strand that will form with it. So now what happened? These all are what? These all are N14 strand DNA. Means what? They are all what? Light DNA. Since I write it with the green, I will write it with the green only. So means what? These all will be what? These all will be light DNA. How many light DNAs are there? 6. Okay. The other is the heavier DNA. Sorry, hybrid DNA. So this is what? N14, N15 DNA, which is called as hybrid DNA. And this hybrid DNA is 2 in number. So if someone will ask you that after 60 minutes, what would be the ratio of hybrid versus light? So after 16 minutes, hybrid versus light. Look in the question, if the light is written in first and the hybrid later on, so you will write like this, or your answer will get wrong. Very important. My question was hybrid versus light. So hybrid is 2, light is 6. You cut it 1:3. But if by mistake you write light first and hybrid later on, this will become 3:1, and in your MCQ that thing will be given to you. So don't get confused. Okay. Now later on what happened, that this thing can have a percentage question, or they can even take you further also. So in your NCRT they are saying that after 80 minutes what will happen? So after 80 minutes you will make like this. Try to make like this. First I want you to do like this, which is a sure shit easy nursery like method. Later on we will do the other method also when we will be practicing these questions for your board and your NEET level examination, so that we can crack it in 1 minute. Clear with this thing? More than this, there will be one more question, that what is the percentage? Okay, how you take the percentage? Now the question I am giving you over here, which we are practicing, is what is the percentage of hybrid DNA after 60 minutes of replication? So when you have to find the percentage, you will take the first the ratio has been taken. Take the ratio. If you don't want to take the ratio, then also it can come, but first we will take the ratio. So ratio is 1:3. 1 is for the hybrid. So write the amount of hybrid on the top. Numerator. In denominator, total. Total would be what? 1 plus 3. That is what? 4. That is the total amount, 4. Okay. Multiply by 100 percentage. So hybrid DNA will be what? This cut, that is 25%. So out of total DNA, 25% is what? Hybrid DNA. This percentage can come in point decimal forms also. Don't get confused that how can DNA will decimal. It's no, they are asking about the percentage. It can come in the decimal also. Clear with this thing? The question, everything has been taught. Later on the plants like Fibabacea, the Taylor did the same experiment and found the same thing, that in plants also semi-conservative nature has been shown. With this, the experimental verification, experimental proof of that DNA is semi-conservative, which has been proposed by the theory of the Watson and Crick, is confirmed. Okay, this is a very important understanding concept. If you understand it, give it a like. If you not, let me know in the comment section. I will help you out with the other way around to talk about it. Moving on to the next thing, that okay, we understood that replication is taking place semi-conservative, but how it's taking place? Still we don't know that how that they two strands, or you separate the two strands, but how? Let's see that in detail in the topic DNA replication. But before going to the DNA replication, that there we need certain of the enzymes which gonna be our superhero for the topics. Let's just start it. Those enzymes which we're going to be requiring, we're going to be learning it one go, so that if you learn it now, later on we will just put it and the process will be keep on doing like makhan. Okay, what is makhan? Butter. Okay, smooth like butter. So let's start. So enzymes, first topic that we're going to be doing for this DNA replication is enzymes for DNA replication. Number one is DNA dependent DNA polymerase. Dependent DNA polymerase. Now, what does DNA dependent DNA polymerase enzyme do? Basically, it's responsible for the polymerization activity, means how the polymerization will take place. It finds out the place where it will start from, means it will find the origin of replication site, from where the recognition of the site, okay, this is the point from where you have to start. Okay, so that thing, what it actually recognized, it recognized the 3' end of the template. You know that we have studied about the DNA segments, that it has a polarity of 5' to 3', and this DNA dependent DNA polymerase find the 3' strand polarity. Okay, so they have to find what? 3'. Okay, on what? On 5' to 3'. Okay, what would be their work like? The work would be formation of 5' to 3' thing. What it is, we will learn it. For now, just remember that they will identify what? 3' end of the strand template. Okay, so it do what? It is responsible for finding the site of replication initiation at, which is present at 3' end of the template strand. Okay, this is it. Then comes second one, that is helicase. Now you know that your DNA is what? In the helical strand, like this. So it has to be opened up. So we will use an enzyme which will open this helix. Okay, so that enzyme is known as what? Helicase. How it will open up? It cut, or it will destroy the hydrogen bonds present between the complementary base pairs. Okay, so it is what? The next one is helicase. It is what? Responsible for separation of two complementary strands from one another by breaking the hydrogen bond. Okay, and this enzyme is responsible for what it will do? Unwinding of the DNA. Okay, makes the DNA unwind or unwinded, opened up. Okay, coming up to the next one. The next is topoisomerase, or it is called as gyrase. Now when we, it's like when we are breaking when we are breaking the hydrogen bond, it is opening up. But when it is opening up, what happened at this location, it creates tension. You might have seen that I always says that when you see the, like in the girls, you have what braids present like this, right? If you will try to open it like this, it will stop, your braid will be stop unwinding. Initially it will unwind for a certain time, after that it will create what, it gets stuck. The girls will able to understand, and boys who don't able to understand, ask your female friends or your siblings, right, or your mother, okay, or any female who has braids. So if you will open it like that, it will get tensed over here, so tension will occur. So basically this topoisomerase and gyrase will do what? It will keep it stretched. Okay, so that it's not getting this tension is removed, so it make a cut over here. So incision is being made by them, topoisomerase, or it is called as gyrase in prokaryotes. Okay, so when they opened up, what happened? It further opened up, like this thing will further open up like a Y-shaped structure. So means later on it's still stuck in the helical form, only this much opened up completely, so it appears like what? Y-shaped structure. Okay, so this is what the role of isomerase. By breaking hydrogen bonds, complementary strands of DNA remain stretched. So topoisomerase enzyme makes an incision / cut at one strand so as to release the tension, and that strand, that will become what? One kind of fragment at one strand. You will understand that thing as we go on. Okay, in this way, Y-shaped replication fork is formed. Okay, so it appears like what? It appears something like this, Y-shaped. Okay, coming up to the next one, fourth one, that is SSB protein, single strand binding proteins, or single strand binding proteins, or helix destabilizing proteins. So SSB full form is what? SSB full form is single stranded, B for binding protein. So what they will do, so they keep the strand in uncoiled form, and the new strand that is being forming, it is helping in coiling it, okay, simultaneously. So this is what its work is. So its work is that prevent reunion of complementary strand, along with that it responsible for coiling of unwinded new strand with older strand. Okay, after this, the next one, we are learning all these different types of enzymes, so that later on we can just work over it, okay, with a process. The next one is DNA polymerase. So what is the role of DNA polymerase? It is responsible for the polymerization process. DNA dependent DNA polymerase is finding and initiating the process. Here it is what? It is responsible for the polymerization of DNA, means keep on adding the new nucleotide in the new strand, responsible for polymerization of DNA. Now in prokaryotes, 3 of them are present, DNA polymerism 1, 2 and 3, whereas in eukaryotes there are 5%, alpha, beta, gamma, delta, epsilon. Okay, so in prokaryotes 3 types present, that is DNA polymerase 1. It is also called as Korenberg's enzyme, just remember it. Okay, then we have DNA polymerase 2, then we have DNA polymerase 3. Now just remember certain facts about them. The DNA polymerase 1, it has what? It do the activity, like major work it do in the activity of the DNA repairing activity. So if they are making the new strand of DNA and it has certain problem, the repair will be done by them in 5' to 3' manner. When I'm saying that 5' to 3' manner, I will let you know what it means. Okay, so it do what? Repairing of DNA in 5' to 3'. When I'm talking about DNA polymerase 2, DNA polymerase exact function is not known, but its function is in 5' to 3'. Exact function unknown, but activity is in 5' to 3'. And DNA polymerase 3, is this the main enzyme which will do the polymerization activity in the 5' to 3', but it also do exonuclease activity in 3' to 5'. When I'm saying that it's doing the exonuclease activity 3' to 5' means what? That it's going in 5' to 3' direction, means new strand forming in 5' to 3' direction, but it's coming back in from 3' to 5' to check whether I have done right thing or not. If it has not done the right thing, it will exclude out the nucleotide, means it will be like kick out the nucleotides which is in the wrong place, which will, how it will do, it will go back. So how you will remember it? Remember that this is a 5' to 3'. Okay, so it's moving like that. Okay, while moving like this 5' to 3', what happened? It in the mid it stops and look back and see back, go back and see that whether everything is correct or not. If it is not correct, it will just exclude the thing out and form the right thing and again keep on moving. Understood this thing? So it's just like you walking and then you check that, okay, whether my friend is with me or not, and you realize that it's left out. So you go back and take it. That is what you do. Same thing. Okay, so this is the one thing. So this is what? It is the main enzyme. It has two functions, polymerization, and polymerization would be in 5' to 3' activity, but it has exonuclease activity, means it is a proofreading kind of a thing in 3' to 5'. Clear with this? Similarly, there are in eukaryotes, and in eukaryotes there are five types, that is alpha, beta, gamma, delta and epsilon, where you have to remember that this one is for the mitochondrial replication, and this delta one is the one used for the nuclear one. Okay, so this is for the nuclear replication. Okay, that you have to remember. Rest polymerization, what the polymerase will do, other alpha, beta and epsilon is not of our concern. Okay, and the last enzyme that we're going to be studying, that is DNA ligase. Now what is the DNA ligase? Ligase word itself means joining together. Okay, so ligase word meaning is what? This word meaning is joining. So basically what happened when the DNA is synthesized, in one DNA would be continuously synthesized, other would be discontinuous with some breaks, means with some cuts. So we have to join those cuts because the DNA is continuous strand, right? So for that we will use it. Okay, we will understand it how that is being formed. Okay, and what those fragments are called as. Those fragments are called as Okazaki fragments. We will learn it, don't worry. So what we say that here we will be just writing it up. Later on you will understand this, don't worry. As DNA is synthesizing, as DNA synthesize, okay, as DNA synthesize continuously on one strand, continuously on leading strand, we call it leading because it's leads, it's don't have any extra work to do. Okay, leading strand, and discontinuously on lagging strand, which forms DNA fragments called Okazaki fragments. Since DNA is continuous molecule, these fragments are joined together. Ligase is what? It's a glue. Okay, so sometimes it is also called as glue, molecular glue also. Okay, so it is also called as molecular glue. Okay, these fragments are joined by DNA ligase to make it what? A regular strand. Now next is the mechanism. So mechanism we're going to be studying in the 6 steps. Here I'm first enumerating the mechanism. Later on we are going one by one to understand it. Okay, so the next thing is mechanism of DNA replication. Mechanism of DNA replication. Number 1, if you want to start something, first you have to see that what is the initiating point. So first what we're going to be doing, we're going to be recognizing the site of origin or the replication of the origin and initiating it. And who do you think will help in it? DNA dependent DNA polymerase, if you would have understood that. So first thing is what? Recognition of ORI and initiation. So recognition of the ORI is what? Origin of replication site. Okay, that is what? Origin of replication site. Then okay, you recognized it. Next thing would be what? Okay, this is winded form, we have to open up to start the process. So unwinding of helix, unwinding of DNA helix, or it is called as DNA duplex. Okay, you have opened it. Next would be to start the process, you have to put the primer first, like the foundation first. So foundation is called as priming. Okay, so first thing is, next thing is you will add the primer called as priming. Okay, so initiating that, putting the lay down the first brick into it. So primer will be there. Then there will be what? Then there will be initiation and polymerization of or the elongation of DNA. Either polymerization or the elongation of DNA. New strand. Okay, then comes the ligation. Means because there will be one segment which will be short, okay, discontinuous. Why discontinuous, we will learn it, okay. Ligation. And the sixth is separation from each other. Finally, completely separated, and the new strand has been formed up with the older one, so semi-conservative nature. So what are the steps? Recognition of the site and initiation, unwinding helix, priming, elongation, initiation of the DNA duplexing, ligation, and the separation. Starting with each step now. So the first step is what? Recognition. Recognition of ORI and initiation. Now what happened in it? OR is the site where the site of replication starts. Who will identify it? DNA dependent DNA polymerase. So in prokaryotic DNA there is only one ORI, but in the eukaryotic DNA there are multiple ORI present. Okay, so that will be identified with a special set of proteins that would be there, and those set of proteins will make certain compounds, okay, that will go and work over there. How they will work, I will tell you. Okay, so what gonna happen? ORI is the site of origin of replication. Means what? For example, if I am making a DNA strand, okay, this is one DNA strand. So maybe this site which I am talking about may be present at this point. So this is what? This is the ORI. From here the initiation will start. Okay, so this is what? Starting point. Okay, so it is what? Starting point. Just an example. Okay, don't take it like big thing, because in the race track also everything comes back to the normal position. So race has to start from certain point, so that is that point only. So this is what? The racing starting point. Okay, I am just writing it in the easiest way possible. In prokaryotes there is only one. In prokaryotic DNA, one ORI only present. But in eukaryotic DNA, multiple ORI is present. And we need certain special types of proteins. Recognizing proteins will help in recognizing it. Certain special recognition proteins helps in finding these sites. And they have very special nucleotide sequence present. Okay, with special nucleotide sequences present of around 100 to 200 base pair. So means they might be present at this level, and then this site is present. Okay, so these sequences are present here. Okay, this thing has been identified by the special proteins. DNA dependent DNA polymerase along with recognizant proteins help in this process. So DNA dependent DNA polymerase along with recognition proteins helps in initiation of process. That's it. Next thing is unwinding of the helix. So who will work over here? Helicase enzyme will work over here. Okay, and simultaneously there will be what? There will be topoisomerase, there will be SSP proteins working over here. Their work, we have done it here, we will understand it in a better way. Okay, so what is happening? The next thing is unwinding of the helix. Unwinding, that is the second step. Unwinding of DNA helix. So we know that who will work over here? Helicase enzyme attaches itself close to the ORI and start breaking the bonds and start breaking the hydrogen bonds between the nitrogenous bases of the complementary strands So what is happening? Let's see that. We have what? We have this thing. Okay, so what is happening here? There must be certain bonds present over here. These bonds are broken up. Okay, so after finding the origin site, these bonds has been broken up like this. And here would be what? Here would be your helic. So this is what? Helicase enzyme. Helicase being present over here. Okay, this helicase. Now the two strands are separated. You can say that one strand is this, other strand is there. Since the strand have polarity, so this will be 5 dash, 3 dash, 3 dash, 5 dash. Okay, it is opened like this. We're gonna be making this diagram only so that we can be remembering the facts, what is happening in our imagination. As these two strands are separated, okay, here what will happen? At this point, tension will develop. Okay, I have to make the helicase little different. Just don't look apt. Helicase would be something like, right now, helicase. They are breaking the bond like this. Not looking better. Here, look. This is how it's breaking the bond. So this is what our helicase. Helicase. So it is a bond breaker enzyme. Now at this point here, what is happening? Something, what? Tension is developed. Tension developed. Now let's take an example. Basically what, for example, think that this is a braided thing. This thing is what? DNA is helix like this. Okay, of course it won't be that good over here, but I will try to show you. For example, this is the braided and this area. If you will try to open it, okay, this is what? This is the 1 point, okay? Just giving you an example, you can imagine it. If you are opening it like this way, okay, the braided, for the boys only this thing is there, girls have understood, right? Let me know, girls, in the comment section whether you understand or not. Okay, so what happened? See this? At this point, I opened this much. Okay, at this point, what happened? It is, I'm not able to open it. Why I'm not able to open it beyond certain level? Because this is creating what? Tensions. Those tensions is what we are talking about over here. This is how I am destroying my braided cables also. But yeah, to let you understand is more important. So who will remove this tension? There will be what? Topoisomerase. Now topoisomerase will do what? The topoisomerase will reduce this tension. Okay, so here what will happen? This topoisomerase will come and will break this tension. How it will break? It will make a cut on it. Okay, so what would be the cut? The cut would be something like, let's see this. This is the strand. Okay, so the cut would be something like this. On one strand it will be cut. So for example, this strand would be cut like this. So as this thing is being cut over here, what will happen? One strand is keep on going, other strand is this. So it opens up a little bit more, like this way it opens up, okay, further. So little bit of nick has been given, incision is given so that this tension could be reduced. It can further opened up, not complete breakup, little bit bond breakups will be there. So this is what, this is the incision given. Incision is what? Certain kind of cut on the surface or on the structure. And this enzyme is what? Topoisomerase. So if I have to write about it, so what I'm going to be writing it up? I'm going to be writing up unwinded DNA duplex develops tension in the unwounded stretch, because complete is won't be opening up now, because that is traveling. This thing is traveling helicase, okay, in the unwounded strand, which is released by, so this is also what, second, which is released by topoisomerase, and in prokaryotes it is called as what, gyrase. Okay, but what it do? By making a cut, small cut, and forming a Y-shaped replication fork, and it forms what? Y-shaped replication fork. Y-shaped replication fork. Now, since this should not go and unwind again, what happened? Certain proteins over here will come. So these are what? SSB proteins. What these SSB proteins do? These SSB proteins keep this prevented from again joining up, because these are what? Complementary pairs. They can again join back. Hydrogen bond is what? As they come closer in the cell, they can join back again. But who is stopping that? This SSB protein. So what is SSB proteins? SSB proteins are like what? They are the what? Protectors kind of a thing of making it go closer. That is what? Moral policing at certain places. Like for example, like Iran's and all those places have what? Moral policing is there. Okay, so this thing is there. SSB proteins prevents the coiling of and rebounding, coiling and rebounding between complementary pairs. But with the new DNA strand that will form on it, it will help in doing it. Complementary strand. With this, the second step is over. Now let's come to the priming. Okay, so I've already written certain points, so we're going to be competing with it, because I made certain mistakes, so I have to come back to this point again after recording everything. So that is what here in the priming, what happened? The DNA dependent DNA polymerase, it will make a primer by what? How it will make? It will synthesize a catalyst. What catalyst will do? That is RNA primer. Okay, it is also called as, it's a type of a RNA strand which work in 5' to 3' direction. Okay, and serves as a function of what? DNA polymerase. More to that, you have to remember that it attaches to the 3' end of the template strand. Whatever be the template strand, whether the dark green covered or light green one which I have shown you, it will go and attach to the 3' end of it. If I'm saying that it will attach to the 3' end of it, it would be something like what? It will be like, so one, this is 3' end. Okay, just remove this. Okay, 3' here, but here if you see that it is what? See here, This is what 5 dash end. So here it can't attached. So how much is open? This much is open. So this is close proximity to the 3 of this strand, darker green color. So the another primer will go and attached over here. So one attached here. So this is what? This is the primer. Attaches at 3 dash polarity of template. Template this. Different template now. Polarity of template strand. Now see how it will work. It will work like this way. So it will form this opposite and this opposite. Opposite to 3 dash is what? 5 dash. And it will make what? 3 dash. So this is what has been told to you, how it will work and what it will form. So primer movement, this is what? It's basically formation, working, activity will be in this direction, forming. But where it goes and join at the 3 dash ends. This is important. If you are able to stick to that, you are able to find which is forming what from here. Okay, so position of primer is different in these two. Now the major question is that one primer I put it here, one primer here. What is the reason? The reason was it goes and join to the 3 dash of individually template. So one template is other, but the activity is 5 dash to 3 dash. This is what the primer topic is. So primer is what? Foundation stone, we have let it. Once that foundation stone is let up, it will start the process. Where it will be let up? 3 dash of the template. So it will find the 3 dash primer and it will go and attach to it. Okay. Moving on to the next thing, then it would be what? Initiation of the process. Okay, so when the process has to be initiated, what will happen? Let's see. Okay, this was our thing. Over here primer was there. So primer will do what? Primer will move. As the primer will initiate the process, soon DNA polymerase will come. Now in prokaryotes, it's gonna be DNA polymerase 3 that will come. And in a eukaryotes, there will be delta polymerase if it is what? If it is nuclear replication, DNA replication. Moving on, what has till now worked? So we know that over here, this is what? This was pink color, right? So this was over here. So it might have moved something over here. Okay, so and it might have added certain things. We are showing it for your understanding. So it is moving this way, right? So it will form, it will forming this whole way. Soon what will happen? It will be, this thing will be removed and it will be covered up by what? It will be covered up by the DNA polymerase. So this is what? DNA polymerase that will come and complete it. In different, it will be different, prokaryotes, eukaryotes. Meanwhile, this will what? It will leave the primer and the DNA dependent DNA polymerase. Simultaneously on the other end also, there is what? It will move, it will form, initiate, but it will also leave. And what will come? DNA polymerase will come. Now, if you see what is happening, DNA replication, you have to remember that this thing is what? These are the nucleotides which are being added up. Now, this process, if you are adding, destroying anything, it requires energy. From where it will be able to get its energy? So the energy will be provided by the already presence of the energies in the system. It won't go and ask mitochondria to make me this much energy. So in the cell, there will be presence of what? There will be presence of ATP, GTP, CTP, TTP. These are what? Triphosphatic form of energy. These triphosphatic forms of energy, what they will do? It will break down. Since the bonds of phosphate which is present with the nucleoside, they are higher energy bonds. So what will happen? When they will break, it will release lots of energy which could be utilized to add the nucleotides in this complementary pairs. First thing. Second thing, this monophosphatic form that will be developed because this, for example, ATP, first form ADP and that AMP. 2 bonds of phosphate has been removed. So it formed what? AMP form of it. This AMP form will act like a substrate for it, because if you remember from the first part of this chapter, you know that AMP is what, ATP is what, these things we have discussed over here. So you can see that those things only we have written out over here, that it has what? Dual functions, acting like an energy source for the whole process. Second, the substrate, the rest left out substrate will act like what? Act as a substrate for the DNA complementary strand. So ATP is there, it will form ADP, released energy, which will be used in the process. ADP again will release one phosphate from its group and form AMP, form monophosphatic form, released energy again. Of course, here more energy, here less energy. So that's why this form. Now the question is that why don't go for the next? Because after that, first of all, energy yield would be very less. So why to go for the process? Second, we can utilize this for the whole process, so means much better utility could be there. Okay, this is about this. Moving on to the next thing. So how the strands are being going further with this process. Okay, so if you see over here, okay, let's just see. And this is, so what happened basically, let's see. Give me a minute. So what we have making, we were making this 3 dash 5 dash 3 dash 5 dash 5 dash 3 dash 3 dash 5 dash. So if you see that at this point, what the helix would have been formed, for example, what from here, this 3 dash, it will be forming like this, and from here it will be forming like this. Can you see that? Here it was going like this way, here it was going like this way. You know this movement. Okay, so this is happening. So this blue color thing which I have shown you is what? That is, I have shown you that this much has been made. Okay, I'm just going to be taking the color. So this reached at this point, whereas this one, it has reached this point. So what will happen? The helix will further opened up. So over here, if you see that they have further added up. Okay, so here what is happening? In this green one, this is present here, this primer, but here, sorry, DNA polymerase. Here DNA polymerase is at this point. So this is what? DNA polymerase. Now as it is opened up, what happened? This is a sequence which is made here, opposite sequence. New complementary strand I'm showing you with what? Blue color. Okay, so this has been formed till here, started from here, as it opened up more. Now the problem is what? The problem arises with this DNA polymerase present on this strand, the strand 3 dash to 5 dash. It says that okay, but this is more opened up, means this has what? Polarity of 3 dash compared to this point. This point has what? 5 dash polarity. Okay, it says that what just happened? 3 dash is still open and over there the fragment is not moved. Then it get what? Crazy, like, oh dude, what I'm doing. So this goes back. It traces back and reaches to this point. And here again start the process. Okay, so what happened? This thing will again go back and again start the process. And this way it came over here again, and this becomes what? Another part. So what is happening over here, if you see this and appreciate this, it becomes what? Two different fragments. Over here it is keep on going continuously, means on this strand it is what? Continuous synthesis. So can I write that this template strand has continuous synthesis on it, means of course complementary to it. That's why we call it what? We call it leading strand. It is the name of the template strand only. Whereas on this template strand has Discontinuous means in fragments, okay, has discontinuous. You understand why it is discontinuous? Because as this DNA polymerase reaches over here, more helix opened up, and it says that why this is not there. Here this is keep on moving, and it's keep on opening. This lighter green color complementary strand will keep on moving, but here it won't be able to do that. It will make a fragments, okay. So has discontinuous synthesis. Hence this strand, template strand, would be called as what? Lagging strand. Okay, these fragments are called as what? Okazaki fragments. Okazaki fragments. Okay, so this is what we have to understand. Now, since these things are not joined together, we have to join it. So the next step that will come is what? That would be the ligation, fifth one. Fifth is ligation. So we're gonna be writing it up. They will be joined together. That's it. Okay. So as DNA replication is discontinuous on lagging strand, to make a regular continuous strand, these Okazaki fragments are joined together by DNA ligase, and this process is called as what? Ligation. Now once they are joined together, the two strands are there. Back of the strand, the new strand that is forming, it has to be continuously recoiled. Who will do that? SSB proteins. Remember it. So that we are not writing it. The last is the separation. Now the two strands has to be separated from each other. So this step, DNA replication, do not stop in the mid of any process. If it has started, it has to be completed. If not completed, then there will be mutation, variations, and so not what. Okay, once they are completely done, the whole of the DNA is completely formed, then only they will separate from each other, and then of course the further process of cell division will take place. If cell divisions do not take place, what will happen? Then there will be more ploidy content will be there. For example, tapetum. Okay, the reasons might be anything, but one such reason we are putting like that they are not, cytokinesis is not occurring over there in the tapetum. Moreover, what else DNA polymerase will do by the rest process is the proofreading and the editing. I told you what, they will go back certain of it and they will check whether the right type of nucleotide is there or not. If something is wrong base pairing is there, any problem is there while copying, things are correct or not, then they will proofread in these steps only. Okay, so that would be what? That is the DNA replication. Do not stop at middle, but completes with replication of entire molecule. Replication of entire molecule. Once replication is over, the daughter cell will separate from one another. Okay, molecule will also separate. Okay, once replication is over, molecules of daughter DNA separate from one another. Each daughter DNA molecule has one parental, old one, that is green or dark green, either of it, and the new one, that is a blue color ones. Okay, so each daughter DNA molecule has one parental, and this is only telling us what semi-conservative nature, right? One parental and one newly synthesized strand. Okay, DNA polymerase itself do proofreading would be better, proofreading and editing. By what way? By exonuclease activity that I have already told you. With this, our all steps of DNA replication is over. We have completed this whole topic in this video. In next video, we're gonna be talking about next process, that is from DNA, how RNA forming and why it is formed. We learned that how the replication of DNA takes place. Since it is being replicated, it is used as a genetic material. So this process is important for you to understand, so that you can later on understand that any problem occurring at this step could lead to lot of mutations in the body, and this is what makes an important category for your medical MBBS, BDS or any journey, to understand that where the problem must have arisen that leads to extra thing in the body. So that's why we are studying over here. I hope you understand that why we are studying this topic for your medical journey. Okay, well, that's all for this video. If you like this video, give it a like, share it with your friends. I know that this is a tougher topic. If you will share, it might be helpful to them if it's helpful to you. And moreover, if you didn't understand anything, you need more crux and the simplest way of remembering everything, I will come up with a short super soon for that. Well, that's all for this video. If you haven't subscribed, do not forget to subscribe it and hit the bell icon, so that whenever I will upload a video, notifications will definitely go within 24 to 48 hours on your devices. Well, that's all for this video. Soon we'll be coming up something very, very interesting for all the students for preparing for the NEET or board examinations from November onwards. Till now, we will be completing our ICSE syllabus on this platform free of cost. Thank you so much.